Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Tick-borne Diseases, Developing Molecular Approaches to Detect Babesiosis, presented by Dr. Robert Molestina. Dr. Molestina is a senior scientist at ATCC. In this presentation, Dr. Molestina will provide an overview of tick-borne disease epidemiology, the current status of diagnostic methods, and resources available from ATCC that advance tick-borne disease research priorities. A special focus will be placed on recent projects aimed at improving the diagnosis of babesiosis via leading-edge technologies. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. First, a quick word about ATCC. Scientific advancements are critical for unlocking incredible breakthroughs in global health. ATCC has provided the credible biomaterials for disease research for over 90 years to help achieve these breakthroughs. We're continuing to cultivate that collaboration among scientists across disciplines by sharing the credible information that is essential for accelerating innovative research that leads to incredible high impact results. Our partnership with you, the scientific community, helps us all to reach the incredible. So with that, I would like to welcome Dr. Molestina.
Thank you, Brian, for the introduction. My name is Robert Molestina. I'm a scientist at the Protostology Laboratory here in ATCC. I'm glad to be with you during this webinar. First, I'm just going to provide an outline of the presentation. So we're going to be talking first on an overview of tick-borne diseases and their impact on the United States. Then we're going to be looking at some of the needs and gaps in tick-borne disease research. This is going to be followed by the types of resources that ATCC offers for tick-borne disease research and diagnostics. And at the last portion of the seminar, we're going to be talking about a couple of projects focusing on babesiosis and a couple of research projects ongoing at ATCC. So first, let's talk about tick-borne diseases in general. So tick-borne diseases are an emerging public health epidemic in the United States. They have become a serious growing threat to public health. Over the last 25 years, there have been reports of Lyme disease in particular that have increased steadily in the country in counties that are now considered to be of high incidence for Lyme disease, increasing by more than 300% in some northeastern states. Scientists have identified a variety of bacterial, parasitic, and viral disease-causing agents that are transmitted to humans by multiple species of ticks. And there are also new tick-borne pathogens that continue to be identified, further implicating ticks as an important threat to human health in the nation. Importantly, as certain species of ticks may have multiple pathogens, people can be infected with more than one tick-borne pathogen at a time. Such co-infections often confound diagnosis and treatment. Currently, the CDC recognizes 13 unique human tick-borne illnesses caused by 18 different pathogens in the United States. So tick-borne diseases in the United States can be of bacterial origin, which include in alphabetical order anaplasmosis, Borrelia miyamotoi disease, ehrlichiosis, Lyme disease, Rocky Mountain spotted fever, rickettsiosis caused by Rickettsia parkeri, tick-borne relapsing fever, and tularemia. Among the parasitic causing agents, we have Babesia microti, which infect red blood cells. And among viral diseases, we have a handful of them: Bovine virus disease, Colorado tick fever, Heartland virus disease, and Powassan virus disease. Those with asterisks are diseases that have to be reported by law to the National Notifiable Diseases Surveillance System at the CDC. The NNDSS helps public health monitor, control, and prevent about 120 different diseases in the United States. This figure shows the total reported cases of confirmed and probable tick-borne diseases from 2004 to 2017. You can see a steadily increase in the last 13 years, with an increase of almost threefold from 2004 to 2017. If we break down the diseases by different categories, we see that Lyme disease continues to be the more frequently reported tick-borne disease in the United States, followed by anaplasmosis, spotted fever, rickettsiosis, babesiosis, and to a lesser extent, tularemia and Powassan virus. You can see this steady increase of Lyme disease, especially between 2016 to 2017. Although it is estimated that there's about 300,000 cases undergoing each year in the United States. Such underreporting is a common phenomenon for most high-incident diseases. Lyme disease in particular, the underreporting is further complicated by a surveillance case definition, which requires both laboratory and supportive clinical data for the confirmation of all but the earliest manifestation of this illness. This graph shows the geographic distribution of key tick-borne diseases in the United States. Each dot represents one case reported from an infected person's county of residence, not necessarily the place where they were infected. Although the graph is from 2015, as mentioned earlier, the distribution of these tick-borne diseases is expanding around the country. It is important to notice an overlap between the distribution of Lyme disease in purple, anaplasmosis in green, and babesiosis in orange along northeastern and midwestern regions of the United States. This goes hand in hand with the geographic distribution of the host vector Ixodes capillaris, also known as black-legged tick or deer tick. And this is shown on the graph on the right. Areas shown in red represent counties in the United States with established distribution of Ixodes capillaris. That is, those counties where ticks were collected in a county within a 12-month period, whereas the areas in yellow represent the estimated distribution of the tick. In addition to the tick-borne diseases mentioned earlier, Ixodes capillaris may also transmit other pathogens, such as Borrelia miyamotoi, Anaplasma phagocytophilum, Ehrlichia murus, Babesia microti, and also Powassan virus. This puts an increasing number of Americans at risk of acquiring other less common Ixodes capillaris associated illnesses. So now that we've gone through a brief introduction of tick-borne diseases and their impact in the United States, let's look at the needs and gaps in tick-borne disease research. Now for this, we need to go back to 2016 and the 21st Century Cures Act. In December of that year, Congress passed the 21st Century Cures Act, which was designed to promote new healthcare innovations for addressing an array of public health issues. Part of the legislation pertained to advancing research on tick-borne diseases. The act required the United States Department of Health and Human Services Secretary to establish a federal advisory committee to review current research efforts and help identify priorities related to tick-borne diseases. In response, the Secretary formed a Tick-borne disease working group to identify gaps in research, education, prevention, and access to care. This group was formed by members of the federal government and the public. Public members included scientists, researchers, physicians, patients, family members, and also patient advocates. To find out more about Tick-borne disease working group, their findings, and a report to Congress, please visit the HHS website at www.hhs.gov. You can also download a complete copy of the report. So based on the findings from the working group, they identified the following needs and gaps in tick-borne disease research. First was to improve early and accurate diagnosis and treatment. Second was to strengthen national surveillance. Third was to understand immunological mechanisms, that is, pathogen-host interactions of immune protection for Lyme disease and other tick-borne diseases. There's also a need to develop new rapid and accurate lab tests, a need to develop antibiotic combinations or therapeutic options for treating acute and persistent illnesses, encourage the development of strategic plans for tick-borne disease federal investments, and also to characterize how these diseases affect United States national security, military readiness, and the health and wellness of active duty service members, veterans, and their families. Once again, to download a complete copy of the report, I will urge you to visit www.hss.gov. So now that we identify the various needs and gaps in tick-borne disease research, ATCC offers a variety of resources for tick-borne disease research and diagnostics. So we go back to the findings of the tick-borne working group, the needs and gaps in tick-borne disease research shown in this table. There are 4 key areas where ATCC can offer resources to support research. That is to improve early and accurate diagnosis and treatment, to better understand immunological mechanisms of immune protection of Lyme disease and other TBDs, to develop new rapid and accurate lab tests, also to develop antibiotic combinations or therapeutic options for treating these diseases. ATCC offers reference microbial strains for tick-borne diseases, other reagents such as antibodies and nucleic acids. We also have the capability to develop assays for diagnostic purposes. ATCC also offers resources to actually study the tick vector. And lastly, as I'm going to be talking a little bit, we also have animal models that represent some of these diseases, mainly rodent models. And these are the different diseases that are represented in our resources. We have resources for anaplasmosis, ehrlichiosis, Lyme disease, rickettsiosis, tularemia, babesiosis, Bourbon virus, Colorado tick fever, and Powassan virus. If you want to find out more about where to find resources in ATCC to help tick-borne disease research, I would urge you to visit our vector-borne disease research website at atcc.org/vectorborne, where you can search for particular organisms or look at our products, as shown in the arrows. You can also click on our ATCC genuine cultures to look for authenticated strains for the different tick-borne diseases. Also, you can search for authenticated genuine nucleic acids, could be RNAs or DNAs for some of these diseases. Or if there's something that is not available in the catalog, you can also visit our customized solutions and look for bulk orders, or probably there could be a nucleic acid that is not offered in the catalog that can actually be made in a custom form for you. I will also urge you to visit our support tab shown here, where you can actually contact us directly with questions, comments, or suggestions about reagents that may not be represented for a particular tick-borne disease that you're working with, or also contact our technical support with suggestions. So those are the different areas where ATCC can offer resources to the researcher who is working on tick-borne diseases. Lastly, in this webinar, I'll be talking about a couple of projects that are undergoing in the ATCC with a special focus on babesiosis. So what is babesiosis? Babesiosis is an emergent tick-borne disease in the United States. It is caused by the parasite Babesia microti. Shown here, we have a cartoon from the CDC representing the life cycle of the parasite. The Babesia microti life cycle involves primarily 2 hosts. These include a rodent, primarily the white-footed mouse, and a tick of the genus Ixodes. During the blood meal, a Babesia-infected tick introduces sporozoites into the mouse host. The sporozoites then enter erythrocytes and undergo the asexual reproduction cycle of the parasite. They can interchange from a trophozoite metabolically active stage to the dividing merozoite stage. Merozoites can exit the red blood cells and reinfect new blood cells, perpetuating the asexual cycle of the parasite. In some cases, the parasite may develop gametes, which, if taken up by another tick, we can have the sexual cycle of the parasite to take place within the tick. This results in the formation of new sporozoites, which can infect another mouse, or accidentally they can transmit the infection by a tick bite to humans. In humans, we again have the asexual stage of the life cycle to take place in infected red blood cells. And also within humans, transmission can occur through pregnancy or blood transfusion, which are less common. Babesiosis is highly prevalent in Northeast and the upper Midwest. Infections among asymptomatic people are mild, and it can cause severe infections in immunocompromised populations. One problem that takes place with babesiosis is in patients who also have Lyme disease. It has been reported that co-infections between Lyme disease and babesiosis can cause worsening of symptoms. And there is a need for new specific and sensitive lab tests for diagnosis and blood screening, which are urgently needed for this disease. So in this area, we have worked into developing new diagnostic approaches to detect Babesia in blood. I'm going to be talking about a couple of them that have been published recently. One of them is the development of quantitative PCR assays for the parasite. It was a study that was published in Experimental Parasitology in 2015. And the second one is a project that has been taking place with collaboration with members of the Center for Applied Proteomics and Molecular Medicine at George Mason University, where we wanted to study the proteomics of Babesia microtia infection in an animal model. So first, let's look at the development of quantitative PCR assays for Babesia. So in this case, we start with the premise that the current gold standard for detection of Babesia microtia infections is a microscopy examination of blood smears. And this can be problematic, as shown in this image on the right, because certain stages of Babesia microtia are very similar to Plasmodium. This blood smear is a mixture of Babesia microtia and Plasmodium bergii, and it's impossible to distinguish the different parasites just by microscopic examination. Antibody detection by serology of babesiosis can provide supportive evidence for diagnosis, but does not reliably distinguish between an active and a prior infection. There have been some recent PCR-based assays, including real-time PCR, developed for Babesia microtia, and has actually been an assay that was recently approved by the FDA last year. When we undertook this project about a few years ago, there was no molecular assays available that will help distinguish between Babesia microtia, which is the more prevalent parasite infection, especially in the northeastern part of the United States, and other agents of human babesiosis that are not as prevalent, such as Babesia duncani, which occurs mostly in the western part of the United States. So in order to distinguish between the species, we targeted the internal transcript spacer regions of nuclear ribosomal RNAs of these two species to develop quantitative PCR assays. This slide shows a representation of the Babesia nuclear ribosomal RNA. Similar to other eukaryotes, it consists of 18S, 5.8S, and 20S regions. These are highly conserved among species. The internal transcribed regions 1 and 2, on the other hand, are divergent, and they help distinguish between species of the same genus. We developed PCRs to amplify ITS regions 1 and 2, and then clone the ampliclone into a plasmid so we could use it later as a molecular standard for quantitative PCR assays. We chose 2 strains of species of Babesia microtia and Babesia duncani, amplify the ITS 1 and 2 regions of these 2 species, and then perform an alignment of these whole regions, and develop primers for quantitative PCR based on the regions on ITS 1 that were more divergent between these 2 species. We came up with the primer shown in red for Babesia microtia and also an internal probe, and the primer shown in green for Babesia duncani, as well as the internal probe for quantitative PCR assays. The next step was to develop these assays using both real-time PCR and also digital droplet PCR to see the sensitivity of detecting the molecular standards, as well as testing these assays in an animal model. The first steps in our approach were to perform serial dilutions of our Babesia standards, shown here in these red boxes. We diluted from 10 to 8 copies down to 1 copy per reaction, and then measure the threshold cycles by real-time PCR. Our assays detected the signals down to 10 copies for each Babesia microti and Babesia duncani, as shown here in blue. And on the right figures, we have essentially the graphical representation of this data. On the y-axis, you have the threshold cycle, and on the x-axis, you have the different copies, dilutions of the copies of the standard. We were able to take down to 10 copies of the molecular standards. We also used our primers in Droplet Digital PCR assays. We diluted the standards from 10 to the 5th down to 1 copy per reaction. And shown in blue is the lowest dilution of standard that were detected by Droplet Digital PCR. And these were down to 10 copies. The figure on the right shows essentially a representation of the data in the table. The blue colors essentially are the different dilutions of the droplets that we're able to detect by ddPCR of the standards. The line shown in pink is essentially the threshold, minimum threshold for droplet positivity, and those in black are essentially the droplets that were negative. So using the assays, we were able to detect again down to 10 copies of the standard by Droplet Digital PCR. We then tested our assays in an animal model. We regularly use hamsters to propagate Babesia microti, Babesia duncani in the lab. We assess parasitemia of hamsters by microscopy, which is a gold standard for detecting babesiosis, and then compare the level of parasitemia by microscopy with quantitative PCR, both real-time PCR and Droplet Digital PCR. This graph here shows the level of parasitemia by microscopy of Babesia microti. We have 2 hamsters, hamster 1 and 2. Early in infection at day 3, the parasitemia is negative by microscopy. We're able to detect parasitemia after 7 days. And if we look at those same samples by quantitative PCR, we see that by Droplet Digital PCR, we got a positive signal as early as 3 days of infection, which was negative by light microscopy. If we look at the other species, Babesia duncani, once again by microscopy, that was undetectable after 3 days of infection. And then by quantitative PCR, we were able to detect the signal for one of the hamsters after 3 days of infection by both real-time PCR and Droplet Digital PCR. So we were glad with these results, although preliminary. We were able to determine that the development of real-time PCR and ddPCR assays were very well established for the detection and discrimination of Babesia microti and Babesia duncani in blood samples. Pending further validation, the assays could be used as highly specific and sensitive molecular tests for screening human blood samples. You can learn more about this particular study and the paper that was published in 2015 in Experimental Parasitology. And also using these assays, we were able then to establish quantitative DNA products that could be used as standards in Babesia qPCR assays that are now available in the ATCC catalog. You can obtain them as ATCC PRA 398DQ for Babesia microti and ATCC PRA 302DQ for Babesia duncani. So having looked at the quantitative PCR assays developed in our laboratory, the next project I wanted to talk about was on the proteomics of Babesia microti infection. And again, this was a study published in 2019 with our collaborators at George Mason University. So first, let's start with this graph. The application of emerging technology in proteomics has contributed significantly to the validation of genome annotation studies in other epicomplexan parasites, which is the phylum where Babesia belongs to, such as Plasmodium and Toxoplasma. The proteomics of research on Babesia, on the other hand, lags behind other epicomplexans. Of note, the great majority of proteomic studies performed in Toxoplasma and Plasmodium have relied on material that's been derived from in vitro cultures. In contrast, the lack of a stable long-term in vitro culture system for Babesia microti poses a significant challenge in analysis of the proteome. So proteomic analysis of Babesia microti is currently restricted to animal models. But there are benefits of analyzing blood and other bodily fluids from such animal models. These include the discovery of new biomarkers of infection, and also the correlation of the progression of infection with the detection of specific biomarkers. But there are challenges in biomarker discovery by proteomics. One of the foremost problems in biomarker measurement is extremely low abundance of candidate markers in blood. Sometimes these exist between the detection limits of mass spectrometry and conventional immunoassays. Such a long abundance will be expected for early stage disease. The second major problem for biomarker discovery and measurement is the overwhelming abundance of resident proteins in the blood, such as albumin and immunoglobulins. These account for more than 90% of circulating plasma proteins, which mask the isolation of rare biomarkers. In fact, the vast majority of low abundance biomarkers are associated with carrier proteins such as albumin. And the third challenge for biomarker measurements is the propensity for low abundance biomarkers to be rapidly degraded by proteases immediately after the blood sample is drawn from the patient. Degradation of these biomarkers can also occur during transportation and storage of blood. So the question is, how can we address these challenges? So our friends at George Mason University have made great progress in the field of nanotechnology, which can now offer new approaches to address these challenges. Our collaborators have engineered hydrogen nanoparticles to support the field of biomarker discovery and proteomics. As shown in this cartoon, these are a nanometer size. They are composed of hydrophilic polymers, which are capable of swelling and contracting as a result of different environmental triggers, such as changes in temperature or pH. These nanoparticles are based on a core which consists of N-isopropyl acrylamide. The core in turn consists of a charged base that performs affinity binding functions for proteins in solution. Here are examples of common baits that are used by our collaborators. Acrylic acid is used as a bait that targets positively charged proteins. Vinyl sulfonic acid, on the other hand, excludes some of the carrier proteins such as albumin. And textile dyes such as Silvercomb blue are useful in the uptake of small proteins on urine. Surrounding the core, there is also a shell that is made of NIPA-M, but it has a variable thickness that can be modified to alter permeability or porosity, such as to exclude large proteins as shown in the cartoon. So you end up with a nanoparticle that achieves three essential functions. The first one is to capture and concentrate dilute amounts of target proteins. The second is to act as a sieving of interferon proteins. And third is to protect the proteins of interest from proteolysis. The proteins sequestered by the particles can then be analyzed by mass spectrometry, Western blotting, and other immunoassays. Our collaborators have used this nanoparticle capture approach in different infectious diseases models. They have found it useful in the capture of antigens of Borrelia burgdorferi in urine. They have also found it useful in the concentration and quantification of another parasite, Trypanosoma cruzi, in the antigens in urine of patients who have co-infections with Trypanosoma cruzi and HIV. And also they have used these nanoparticles as a recent proof of concept study showing that Toxoplasma gondii antigen detection in urine is possible in rodent models. And they did this by use an appropriate affinity concentration method before doing a mass spectrometry analysis to identify proteins of Toxoplasma. So as nanotechnology methods and mass spectrometry instrumentation have advanced sufficiently to allow protein discovery directly in complex biological fluids, we collaborated with the personnel at George Mason University to examine the Babesia microti chromium in whole blood. We wanted to determine the capability of the nanoparticles to sequester proteins of the parasite using our hamster model of babesiosis. We then wanted to identify the captured proteins by mass spectrometry and also present a preliminary description of the Babesia microti proteome to complement previous genomic and transcriptomic studies. And lastly, one of our hopes is in the future to contribute to the development of new diagnostic tools as we are able to identify new biomarkers of infection. This is the experimental strategy that we followed in this project. First, whole blood is collected from experimentally infected hamsters with have greater than 30% parasitemia. In this case, we use Babesia microtia G1, also known as ATCC PRA398. Then the erythrocytes are lysed, and hydrogen nanoparticles are used to sequester parasite proteins and also help exclude high abundance proteins from the host, such as albumin. The proteins are then enriched, eluted from the nanoparticles. And lastly, mass spectrometry is performed using an LTQ fusion mass spectrometer. The tandem and mass spectra obtained from this analysis are then searched against the Babesia protein database. The cutoff peptide length in our analysis is 7 amino acids, and any peptide that shows similarity or greater than 80% to the host golden hamster or any other protein in an unredundant NCBI database are rejected. So these are the results that we got from this study. This pie chart shows the different functions of the proteins that were identified in analysis. We were able to identify greater than 500 Babesia proteins in whole blood of hamsters. Nearly 130 proteins match to hypothetical genes. About half of those hypothetical genes belong to conserved proteins in Plasmodium falciparum. The majority of proteins identified also included ribosomal proteins. We also able to detect transporters, proteins involving glycolysis, the TCA cycle, chaperones, proteases, proteins involving trafficking, and also other examples included proteins involved in cell signaling, lipid metabolism, surface and or secreted proteins such as members of the Babesia BMM family. The purpose of the table is to give you a sense of the diversity of parasite proteins that were captured in the nanoparticles based on their abundance in the Babesia microtia proteome. These are listed in order based on abundance. Proteins that have a higher PSM or peptide spectrum match are those that are found on top of the table. We detected many of these surface secreted antigens from the BMN family of proteins, which are highly abundant in the Babesia microtia proteome. And these are good targets to develop diagnostic assays because previous studies have shown that they trigger robust immune responses in the host. We're also able to detect a number of heat shock proteins that's really highly abundant in the proteome of Babesia microtia. So the next step to do is, of course, to try to validate some of the findings of our proteomic study. This table shows the utility of using the nanoparticles and trapping different surface or secreted Babesia antigens, which previous studies have shown to elicit host immune responses in vivo. Many of these antigens included GPI anchored, secreted, and transmembrane proteins, some of which share homologies to Plasmodium falciparum. Many of these identified seroreactive antigens are among the top 10% most expressed genes in Babesia microtia, as shown by previous transcriptome studies. Once again, the one at the top of the list is BMSA1. We develop polyclonal antibodies against this protein to ensure that it is expressed in infected red blood cells. The image shown on the right is an immunofluorescence assay with antibodies to BMSA1, shown in green. Blue is essentially the nuclei of the parasite stained by DAPI in infected red blood cells. And on the bottom, you have a merged image of the light microscopic image and the green and the blue as shown with the arrows. So localization of this particular protein is shown around the trophozoite stage of the parasite and also staining within the cytoplasm of the red blood cell. We also develop an antibody to a second antigen, BMRI1 3G00947. The localization of this particular protein was very interesting. It actually shows a diffuse presence on infected red blood cells as shown here. So we'd like to continue developing antibodies to the next set of antigens that we identified proteomic studies and hopefully test them in new diagnostic assays for babesiosis in our animal model. So as a summary from our proteomic studies, we presented a partial description of the Babesia microtic proteome by successfully capturing and identifying parasite proteins from whole blood using a combination of nanotechnology and mass spectrometry. The catalog of proteins identified mirrors the metabolically active trophozoite stage. We found that surface-secreted antigens are highly abundant in the Babesia microtic proteome and serve as useful biomarkers of infection. The approach we use in the study avoids the cumbersome separation of infected erythrocytes and free parasites from the sample. And the molecular sieve and affinity functions minimizes the capture of highly abundant interferon proteins from the host. If you'd like to learn more about this particular study, I will invite you to look at our paper published in International Journal of Parasitology in January of this year. So in conclusions, I'd just like to mention again, we saw that diseases transmitted by ticks are a serious and growing public health concern in the United States. There still remain several gaps in tick-borne disease research, education, prevention, and treatment. ATCC offers a variety of resources that are critical to advancing tick-borne disease research priorities. And we talked about an ongoing research project in ATCC that are focused on improving diagnostic methods of babesiosis. That brings me to the end of the seminar. If you have any questions, please let us know at the end. And I switch my word to Brian for any final comments.
Well, thank you, Robert. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org.